So it is my great delight to be here. I'm very excited to now be an adjunct member of the department and to be talking today about genes, microbes, and how they influence brain development in infancy and early childhood. So I wanted to start out by kind of framing the challenge that my research program was designed to address. And the challenge is that by the time the clinical symptoms of many neurodevelopmental and psychiatric disorders emerge, whether it's autism, schizophrenia, intellectual disabilities, a lot of the underlying atypical brain development has already happened and may be very difficult to reverse at that point in time. So I really do think that our best hope of making an impact on these conditions is to focus on prevention and very early intervention. And keeping that in mind, the mission of our lab in the long term is to develop therapeutic interventions that could actually adjust atypical neurodevelopmental trajectories early in life. But to do that in the short term, first we have to learn how to identify at-risk populations in infancy, so you actually know who you can intervene with. And second, we need to understand the genes and molecular pathways that shape neurodevelopment in these key periods. Because that's going to give us what our targets might be. The primary approach my lab uses is something called population neuroscience. This is a concept originally defined by Thomas House, who said it was a branch of neuroscience that seeks to identify genetic and environmental factors that shape individual differences in human brain structure and function using state-of-the-art methods in genetics, epidemiology, and cognitive neuroscience. So I often like to think about it as the integration of genome, envirome, and phenome. Now, when it comes to the genome, it's a very large search space, but it is thankfully a finite one. And we have a lot of tools to probe it. A lot of my work focuses on genome-wide association studies, so using arrays to look at single nucleotide polymorphism, so common variation and its impact on risk and development. But we also do some work looking at copy number variants, including chromosomal aneuploidies. We do some work with infants and women with Turner syndrome. And we also do a little exome sequencing work that we've gotten into recently, where we can look at rare variants as well as insertions and deletions. In terms of the phenome, a lot of what I do is neuroimaging, particularly MRI-based neuroimaging of various types. But we also do behavioral and cognitive assessments with our participants. And we do some analytical chemistry, for example, looking at hormonal responses to stress or looking at neurotransmitter metabolism. Right, so what is GWAS? Probably this room does not need this definition. But basically, this is using microarray technology to identify associations between specific outcomes, height, IQ, schizophrenia, in our case, things like brain volumes, white matter microstructure, and genetic variants, looking across the entire genome rather than confining yourself to a specific gene based on what is probably inadequate a priori knowledge. So this is really a talk of two halves. So I'm going to tell you a story about genes. Oh, you know what? I completely skipped environment. You know, some days that starts to throw me off. Let me talk about the environment for a minute. So the environment is actually a lot more difficult than the genome, right? Because it's an essentially infinite search space. And there are many, many different things that we could be looking at. So my lab has particular interest in commensal microbes. And the second half of my stories today will be a story about the gut microbiome. We're very interested in sex hormones as well. But I won't talk about that today. And we recently received a new grant to actually look at psychosocial stress and environmental chemicals and how they move through the microbiome and also inflammatory markers to influence development. So apologies for the brief recursive section there. So I want to show you the first genome-wide association study that we conducted and that was ever conducted on an imaging phenotype in infancy. So in this study, does this have a blinky? It does. We were looking at global brain tissue volumes, so intracranial volume, as well as gray matter here in the green, and yellow and red is white matter, blue is CSF. This was leveraging an ongoing prospective longitudinal pregnancy cohort study from the University of North Carolina at Chapel Hill, where I was for 13 years, run by a gentleman named John Gilmore. And in addition to getting the brain scans, a large number of the families donated buccal samples for genotyping. Now, it's relatively small as a GWAS. It's 561 neonates with complete data, both high-quality imaging and high-quality genetics, and over 9 million SNPs post-imputation. Now, despite the relatively small sample size, this is really a proof of concept study, we were excited to see that, in fact, there was evidence that common genetic variation influenced global brain volumes. And in particular, we found a genome-wide significant locus for global gray matter volumes. And you can see that the locus plot on the top of the middle column here. And this is an intronic SNP in a gene called IGFBP7, which has been implicated in learning and memory. But perhaps even more interestingly, it's within 500 kb of a gene called REST. And REST is known to be a master negative regulator of neurogenesis. So basically, it turns off neuronal genes in non-neuronal cell lineages. So it's really important. We also had a locus that near genome-wide significance for white matter volumes. And this locus is in a gene called WWOX, which in the adult human brain has been looked at in younger brains. But the adult human brain is exclusively expressed in the axons. So it makes sense for a white matter phenotype. There are cases of people with protein killing mutations in this gene, and it is associated with discernible differences on MRI, including things like white matter hypoplasia. So we were excited. Now, normally, if you did a genome-wide association study, you'd do a replication cohort. But because we were dealing with a phenotype that's difficult and expensive to get, there wasn't an opportunity to do that for this initial study. So we did some other things to look at the biological plausibility of the findings. One was to look at gene expression levels. This is data from the BrainCloud database, looking at prefrontal cortex gene expression. And you can see up here that IGFBP7 shows a strong increase in expression across gestation and early life, and then sort of stable through later childhood and adulthood. REST down here, as you might expect, very highly expressed during neurogenesis, early in gestation, and then reduces over time. WWX is interesting because it depends on which probe you use. So one of the probes that BrainCloud used shows a strong increase over development. The other shows elevated prenatal expression, suggesting that WWOX might actually have different isoforms that perform different roles at different points in development. We also looked that if we moved beyond the top hits and looked at sort of more liberal p-value thresholds, how those groups of genes were expressed across the lifespan. And what we saw was that the GWAS associations across multiple different cutoffs appeared to be enriched for genes with elevated prenatal expression. In keeping with our phenotypes, these kids are all scanned about the first month of life. And in fact, you can see it's actually strongest for the highest p-value thresholds. We also did a follow-up study looking at white matter microstructure. So white matter microstructure, I'll talk a little bit about. The white matter tracks are really like the telephone wires of the brain, right? They're connecting up different brain regions and allowing them to communicate with each other, even if they're spatially distant. Diffusion tensor imaging is an MRI technique that allows you to reconstruct those pathways using the diffusion of water in the brain. So it's very cool. You get these beautiful pictures, like the one over there on the right, showing major association tracks in the brain. And then you can take diffusivity indices of different types from these images. A common one is fractional anisotropy. That's what I'll be showing you today, which basically tells you how directional the diffusion is. In white matter, it's very directional because you've got the axons packed together, you've got myelin around them. So it keeps water from moving. But it also is a marker of integrity and maturation. Typically increases over time. So same cohort of kids, but now with a different phenotype. And you can see here a DTI scan where basically they lit up the directional movement of the track in different colors. So here you see, right, the bright green is a track moving across the brain, it's the corpus callosum. Blues are moving backwards and forwards. Now, one of the big challenges in doing imaging genetics is imaging phenotypes tend to be very complex and multivariate. So I already showed you, right, that picture of multiple association tracks. We actually have 47 tracks in total in our brain atlas, and you can see them all here. And in addition, if you take any single tract, you kind of see this in a little dark, but you can look at those diffusivity indices across every kind of cross-sectional place on that arc length. So you could have, hundreds and thousands of phenotypes. You need a really large sample size to control for that. So what we did is we extracted a latent measure of white matter microstructure from our FA values across all of these tracks. I think this is really interesting. You can see here the covariance matrix of 5 principal components extracted from each tract. And you'll see that the first principal component of all the different tracks are highly correlated with each other. So this is our common factor. It explains about 50% of variation across all the tracks. You can see in the middle one, it explains a large proportion of variance within each individual tract, with a few exceptions right there at the bottom of the plot. And interestingly, it accounts for a large portion of the heritable variation in the tracks. So the EBDS study actually has a subset of twins, so we can actually do classic twin-based case modeling. And what you can see here in the circle plot is the outside is additive genetic variation for each tract individually, and here for our common factor. And the inside is showing what happens when you control for the common factor. So you see heritability drops for all the individual tracks once you take the latent factor into account. We were excited to find that we did have, see, there we go, a genome-wide significant hit for the latent white matter microstructure factor. It's a gene called PSMF1, which encodes a protein in the 26S proteasome. And this particular complex is known to play a role in developmental axonal pruning, as well as synaptic plasticity. So it did look interesting biologically. This is sort of all the kind of top hits, things that are suggestive or above. And what's interesting is, a theme kind of emerges. Oh, that red is so dark. I do apologize. But you can see that multiple of these genes are involved in axonal pathfinding and fasciculation. One's involved in myelination as well, but most of them tap earlier developmental processes. And the SETBP1 is also very interesting because it's a high confidence autism associated gene. We also integrated our results with existing gene expression databases. And you can see that for our top hits, a number of them are expressed in fetal brain rather strongly, whether you're looking at protein or gene expression. And if you look at change over time, you can see a subset of these genes show that pattern of elevated prenatal expression in the bright red and then shifting over time. So less expression later in development. What we don't see, however, is if we start looking at these more liberal p-value thresholds, we don't see an enrichment for genes with elevated prenatal expression. It looks different than the volume data. And I suspect that's because a lot of these white matter development processes are happening later in gestation, where we don't have as much data available. The prenatal expression data is strongly driven by early development. Because we have all this individual beautiful tracks, we wanted to look at them as well. So in most cases, the loci that we were examining showed significant associations with FA in multiple fiber tracks and across the entire bundles. However, there are some tracks, this includes the right and left optic tract and the left and right medial meniscus and superior portions of the left and right cingulum, which show relatively weak associations with the variants we examined. We also found that some variants exert very strong effects, but only in a subset of tracks. So find it here. So there's a gene called CNIH3, which appears to be particularly important for the development of the arcuate conspicuous. So you can see that kind of highlighted there, which is involved in language processing. Oop, that didn't do what I expected it to do. Did something. There we go. And the studies in rodents actually show that this is related to the trafficking gaining properties of AMPA, selective glutamate receptors. There we go. So this is actually kind of an example looking at local effects of that top SNP across all the different tracks. So what you'll notice, of course, is that most of them, the red is highly significant here, show an association with the SNP. But there are a few areas that don't, these dark blue ones. And that's particularly down here as we go into the brainstem, and a little bit in these kind of central areas of around the thalamus. And these are the very areas that myelinate early in life. So I think what's happening is that the latent variable we're picking up is tapping kind of mid-gestation or late gestation developmental processes. It's not tapping into myelination, primarily a postnatal process. And if we were to rerun the study with data from 1 or 2-year-olds or older children, it would look quite different, probably a different set of genes. And you can see that when you're looking here too. This is the corpus callosum. You can see that significant effect size drops in the middle of the tract where it myelinates first. And the same thing if you look at these corticofugal tracts that connect the cortex to the spinal cord, you can see that significance and effect sizes drop as you get deeper into the brain. So quick conclusions for part 1. We discovered that common genetic variation influences global tissue volumes and white matter microstructure in infancy. Many of the individual loci identified actually have a clear relevance to neurodevelopmental processes, intellectual disability, and mental illness, particularly in the volume study. A number of the top hits had also appeared in GWAS of things like schizophrenia, but independent replication in a larger sample and a more diverse sample is needed. And I'll get back to that at the end in future lectures. But now for something completely different. Talk a little bit about microbiomes. So in a sense, this is genetics, but now we're going to talk about the genes of our microbial companions in our gut. So what is a microbiome? Microbiome is just an ecological community of organisms that are living in a particular environment. They are commensal, which means they're living in close association with both each other and us. Most of them are thought to be symbiotic, at least with the host, that we help each other out. Sometimes they're symbiotic with each other, sometimes they're antagonistic with each other. And some of them may be pathogenic, right? Capable of actually causing disease, particularly under certain circumstances. If we want to be picky about it, people will often say that microbiome specifically refers to the combined genetic material of these organisms. I'm going to talk a lot about bacteria today, but there are also viruses, fungi, and protozoa in this community. And what was really exciting for me as a fairly young scientist when I started looking at it is that these trillions of microbes actually appear to influence brain development, cognition, and risk for mental illness. So a lot of this comes from rodent studies, and particularly germ-free animals, where if you take away their typical microbiomes, you see drops in recognition memory, reduced sociability, reduced anxiety, which you might think is a good thing. In humans, we think of anxiety as bad usually. But if you're a small prey animal, maybe you should be scared of the world. So you can think of this as a path of pathological change. That's accompanied by changes in brain chemistry. So BDNF levels drop, levels of serotonin, ACTH, and corticosterone go up. And there are complex changes in neuronal transcription and neurotransmission. And I should say there's also increased locomotion and self-grooming behavior in these animals. So why? Why would that happen? How do things in the gut talk to the brain, which is so physically distant from them? And there are a lot of different interesting pathways. So one, the microbiomes interact with these enterochromaffin cells in the lining of the gut, which have a very important role in producing peripheral serotonin. So they can actually change the production of 5-HT. They also interface with the immune system, both the intestinal immune system and the systemic immune system. So they can trigger inflammation, and those inflammatory cytokines can pass into the brain and change how the brain functions. They also produce a bunch of really interesting molecules that are actually neuroactive, including the short-chain fatty acids, which again are capable of passing the blood-brain barrier, so could actually change gene transcription in the brain. And then they make classic neurotransmitters like dopamine and GABA. I suspect those aren't getting all the way to your brain, but what they can do is they can diffuse through the lining and impact signaling through the vagus nerve, which is basically your one-stop route to your brain. Which is there, so that's indicating the base. So we were really interested in whether these things that have been observed in animals would be observed in humans, and particularly in infancy, because it's a key period in brain development, and it's also when the microbiome is being established. So our first study to look at this was looking at infant cognition. We were leveraging that same EBDS study we used for the genetic work, but with a reduced sample size because it's added right on at the end. John Gilmore gets the head. Andrea S. Perati-Carril runs the microbiome for UNC, so she ran all these samples and did the bioinformatics with her team. I'm going to show you functional connectivity data that our colleague Wei Gao produced, and Barbara Goldman here, since retired, did our cognitive assessments, which are done with the Mullen scales of early learning. And this was supported by the Foundation of Hope, in addition to the NIH. So what we observed was that infants can be sorted into 3 clusters based on the composition of their 12-month microbiome. One of these groups is characterized by relatively high levels of something called Faecalibacterium, which is more typical of adult microbiomes. This may be a more mature community. The middle group is characterized by high levels of Bacteroides, which is a more juvenile type microbe, although you do see adults who have high levels of Bacteroides as well. And the third one is high levels of this Ruminococcacei, which I know I butcher the pronunciation every time, but I try. Most exciting that we saw is that both cluster membership and alpha diversity, I'll explain what alpha diversity is in a minute, were associated with cognitive performance by these kids. And in particular, cluster 2, that high Bacteroides cluster, performs better than the other 2. None of these kids are cognitively delayed. They're all performing the normal range. But this is a difference between being around the 70th percentile and the 90th percentile. So it is not an insubstantial group difference. Alpha diversity is basically a measure of how much diversity is in any individual sample. So you see one of these measures here is observed species in the middle. That's kind of the easiest measure to understand. If you have more species, you have a more diverse microbiome. Some of these measures also take account of things like how much of really rare groups you have. That's what Chow-1 is on the left. And then phase phylogenetic diversity takes account of how closely related they are. So if you have 2 microbes in your gut, oversimplified scenario, and they're closely related, that's less diverse than if you had 2 distantly related ones. But regardless, for all of the measures, the more diverse microbiomes were associated with poorer cognitive performance. And that was surprising. Historically, alpha diversity has generally been considered a good thing. You have more microbes, you have more redundancy in functional pathways. If something gets knocked out by a disease or antibiotics, somebody else can come and fill in. But we see the opposite. One possibility is that diverse microbes, microbiomes in this age, are harboring more pathobiomes. Another possibility is that there's just a lot more competition here, and therefore the beneficial microbes are getting less of the resources they would need to really make a difference on the system. And perhaps most intriguingly, given that it's the juvenile cluster that performs best and that alpha diversity increases with age, maybe there's a cognitive benefit to having slower development of the microbiome. And that's something we're really interested in exploring in our new work. We also see that alpha diversity associates with functional connectivity in the brain. And in particular, connectivity between sensory motor areas and the left inferior parietal cortex. And this was really exciting because not only does alpha diversity correlate with this, but this measure also correlates with cognition. So it looks like this is a neural circuit that mediates microbiome associations with cognitive outcomes. The IPL is involved in top-down attentional allocation and social cognition and language. So when you think about a baby learning, those are kind of really key areas, that and the sensory motor, because they're learning through touching, through sticking things in their mouth. So you can see how that might relate to cognitive development. Based on the rodent literature, we were also very interested in anxiety and anxiety-related phenotypes. So we looked at some other connectivity differences, and we see that the anterior cingulate cortex, which plays a key role in emotional regulation, and its connectivity to the anterior insula correlated with alpha diversity. We also saw the connectivity between the amygdala, a really key structure in processing threat, and an area of the thalamus near the ventricles, which we also know is very important for fear processing and negative emotionality, was associated with alpha diversity. But we didn't have any behavior that was anxiety related in this kid. That's not what ABDS was designed to look at in these ages. So we got a new grant from the NIH to look specifically at anxiety related behaviors in infants. Kids came in at 2 weeks and 1 year of age. We got poop samples, we got saliva, we got all the good biosamples, we got blood spots. The saliva is collected before and after blood spots, so we could look at hormonal responses to an adverse stimulus. The blood spots are still on ice, but will soon be processed by the mass spec core here, and of course MRI. Then when they came back at 1 year, they did all of that, but also did these two tasks, the strange situation and the mask task, that I'll show you in a minute. We also did cardiac recordings during these. So some of the same faces here, but I want to give a shout out to Karen Gruen, who advised us on the cortisol reactivity measures, Martin Steiner, who analyzed our structural MRI data, Kathy Proper, who advised us on these behavioral paradigms, and graduate student Alexander Carlson, who did the paper on the mask task in strange situation, and Samuel Rosen, who did a paper on the cortisol reactivity, and Kaija did our statistical analysis. All right, so now we get to see if the videos work. All right, so this beautiful little girl is doing the strange situation task. In this task, the mom is sitting in a chair in the room. The kid is in a space they haven't been before. They've got lots of new toys to play with. And after a couple of minutes of acclimation, a research assistant they haven't met before comes into the room. At first, the assistant just sits in a chair opposite from the mom, quietly. Then after a couple of minutes, they start talking with the mom. And then after a few more minutes, you can see Alex coming in there. They actually get down on the floor and start trying to interact with the baby. So it's kind of an increasing level of social intensity, maybe. So you can see this little girl has basically frozen the minute she sees Alex, which is actually really unusual for babies. And she stays frozen for 30 seconds up to a minute. And then she actually starts crying a little bit, even though Alex has literally done nothing at this point. He hasn't even tried to interact with the mom or her. That's actually a pretty intense social weariness in a kid this age. This little boy here is doing the mask task. The mask task, they're in the high chair. You see the white curtain there. The research assistant is behind the white curtain. He's going to put on a Halloween mask. Then he comes out and he says the child's name a couple times, then goes back behind the curtain, waits a little bit, and does another mask. Cheerios. So we don't have the volume on, otherwise you would hear this little guy, not a big fan of the apple mask, does not like that one. He seems so surprised. How has this happened to me in my life? What have I done? But I think this is so fascinating. Obviously, these were chosen to show extremes of behavior. Many, many children do not react that way to the mask. Some of them, we show up to four masks. If a kid has a really strong negative reaction to the first one, and it's hard crying more than 15 seconds, we stop the paradigm then. Because we've already seen that they are the most intense responders. And they're not, we'll go on and show them some additional masks. And some kids will be smiling and laughing all the way to the end. He's not. The other thing I think really interesting about him is as soon as the mask is gone, he comes right back down. So some kids get riled up and they stay riled up. But he seems to actually have pretty good emotional regulation. And that's something we're interested in looking at more in the future. But what we looked at first was we looked at cortisol reactivity to that heel prick. This was Sam's paper in psychoneuroendocrinology. You can see that in the neonates, most children have a pretty strong cortisol response to a heel prick. And what we saw was that this was associated with alpha diversity. So the kids with more alpha diversity, which you'll recall in our previous work was associated with lower cognition, also have higher cortisol responses. So they seem to have a stronger hormonal reactivity. We didn't see any associations with the strange situation task with the social kind of awareness paradigm. For the mask task, which I think is probably tapping into an older neural circuit that's dealing more with really immediate stressors, like maybe predator stressors. We do see associations. First, we see that alpha diversity at 1 month actually predicts reactivity a year later when we do the mask task. Again, the higher diversity kids are the kids with stronger behavioral responses, as they had stronger hormonal responses. And then we see that the composition of the 1-year microbiome is associated with concurrent theory activity. So this little taxa bar plot here, each column is a child, and the colors show the relative abundance of different bacterial groups. And this is organized by the first principal coordinate. Microbiome data is also highly multivariate, so we do a lot of things trying to reduce the data and summarize it. So you can see as you get to the right, you've got kids with more of this gray group in general, that's Bacteroides. And you see reduced levels of kind of this medium blue group over here, which is Veillonella, Bifidobacterium, which is dark blue down here on the bottom, dark blue down there, and Lactobacillus. So these kids, so the kids who are scoring high on this are showing reduced fear reactivity in the mask task. Interestingly, we also see that there are correlations between gut microbiome features and brain volumes. We look specifically at amygdala, hippocampus, and medial prefrontal cortex, very important for fear processing and anxiety. So we can see that the 1 month principal coordinate in this case, so now we're looking at the beta diversity, is associated with medial prefrontal cortex volumes a year later. And we see, I thought this was the most exciting thing, is that the same principal coordinate that is associated with behavioral reactivity is associated with amygdala volumes at that age. So we're not really powered to do a proper mediation analysis here, but it looks like this might be, again, a neuroimaging phenotype mediating between the microbiome and behavior. I'm going to end the review of the existing data with a little move out of epidemiology and population neuroscience into some preclinical studies. So this was something I was able to add to my lab when I moved to MSU. But of course, the question of the human studies is, are these associations causal? It could be reverse causation. Maybe kids who are anxious, that changes their gut motility, and that changes the kind of microbiome that they can support. Or maybe it's just serving as a proxy for some other factor. And I should say, we try to control for a lot of the known factors like breastfeeding and delivery style. But really, we know very little about what drives microbial diversity in these ages. Most of it is unexplained. So what we wanted to do was to take microbiomes from our kids and put them into germ-free mice and see if we could recapitulate some of the phenotypes. So this is a little picture of kind of the outline of the experiment. These are germ-free animals at the University of Michigan Germ-Free Core. They got the microbiomes inoculated around 10 days of pregnancy because we wanted the infants to be exposed right from birth and on. My graduate student, Antonio White, is actually looking at how this might affect maternal behavior in the different groups. And then at about 6 weeks of age, they start doing a range of different behavioral tests that target anxiety, depression type phenotypes that our kids were too young for us to look at, but could become important later, as well as some things like the Carmen Red test, which tells you about gut motility, which again, we weren't able to look at that directly in our kids. And then they had neuroimaging and were sacrificed, and we could do some post-mortem work. Again, not possible in the children. So Harikish Dubey has been leading this as postdoc in my lab. Sheng Lu is our lab manager, so he handles a lot of day-to-day operations. Christiane Millette is the head of our molecular imaging core at the IQ. So she assisted us doing neuroimaging with animals, which we had not done before. Turned out to be a lot more complicated than doing humans. Rohan Rahachuri is a osteopathic medicine student. So he's been doing our dendritic complexity measures with the postmortem samples. And Charlotte Best is a talented undergrad from the professorial assistantship program originally. She's now been in my lab 3 years. But she's been doing myelin measures with electron microscopy. We made 4 different groups of animals. One of them got a humanized microbiome that was rich in bifidobacterium. One of them got a human microbiome that was rich in Bacteroides, so some of the key players in driving variation in these ages. Then there was a group that got like normal mouse SBF microbiomes, but still went through the manipulations. And then there were germ-free animals that got sterilized communities. So basically they got dead bacteria, not live communities. And we've seen some interesting things. So if you look at the top one here, Male SPF mice spend more time with a familiar mouse in a social test than either the male bifidobacterium mice or the back mice. So it seems like just being a humanized mouse, maybe of any kind, changes your social behavior and takes away kind of we expect to see social novelty in mice, like they like meeting new mice more than they like hanging out with their old mice friends. They're just like people. This is the corticosterone response to a restraint stress. So this kind of mirrors our heel prick and cortisol response in the kids. And what we see is that the germ-free mice show a bigger increase in corticosterone. They also have lower basal levels, which is something that's been reported in these mice before. And then if you look at the SPF mice, they return to baseline faster than any of the other groups. The other groups stay elevated past the typical end of the test. So again, interesting, but it doesn't tell us much about the difference between the two human groups. Where we saw a difference between the human groups was for dendritic complexity. So both the volume and the length of the dendrites, as well as the synaptic density on the dendrites, differs such that the BIF mice are similar to SPF mice. So they don't show negative effects of being humanized. Whereas the back mice and the GF mice have reduced complexity. And this is in prefrontal cortex, amygdala, hippocampus, nucleus accumbens. Looks very similar. So conclusions for part 2. We've discovered that certain features of the human infant gut microbiome are associated with its cognitive development, with their behavioral responses to potential threat, their functional brain connectivity. The previous literature suggests that infants with mild cognitive impairments, dysregulated HPA axis function, and or extremely strong negative reactions to these kind of stimuli are at increased risk for later problems. So that suggests that the composition of the microbiome may be an early risk or protection factor that could be manipulated to change long-term outcomes. One of our goals in making the mice was we would have a preclinical model to try out some interventions. The challenge, of course, is we still don't know very much about the mechanisms by which these specific microbes or microbial communities might influence infant brain development. And that's another reason for having those mouse models. I'll give you a little heads up for new things coming down the line. In terms of genetics, about 1.5 years ago, we received a large grant from the NIH to create the largest and most diverse imaging data set ever focused on infancy and early childhood. So we can address that problem of small sample size and lack of replication data sets and lack of diversity. So this has been done through the Organization for Imaging Genomics in Infancy, which I founded in 2017 to facilitate this kind of work. It's now grown to include 21 different cohorts coming from 17 different institutions. And ultimately, we're expecting to have a data set of more than 6,000 children, with most of them having multiple brain scans and a significant subset having detailed behavioral and cognitive data as well. So and it is a more diverse sample. There are a lot of US sites, but we also have sites in Germany, the UK. We're working with FinBrain now to bring in their data. Cape Town, South Africa, as well as Singapore. So that's exciting. In the microbiome, my colleague Kathy Proper at UNC and I have an MPI grant to look at how early toxic stress influences the development of the microbiome in young children and its relation to anxiety-related behaviors. So it's like an older sample than the one I showed you, but similar kinds of paradigms. And then I received an NIH grant just a couple months ago. Nicole Talgi, who's with us today, is part of the co-investigator team for that, to look at environmental chemical exposures and psychosocial stress during pregnancy, and whether the microbiome and inflammatory profiles mediate the impact on neurodevelopment measured around 4 years of age. I have not talked at all about sex today, but it is something that I'm very interested in. So we're using some startup and discretionary funds to look at genetic variants that might be linked to social cognition, executive function, and visual spatial problems in women with Turner syndrome, using exome sequencing data. And then Aunt Alex, one of my other postdocs, very talented, is looking at the genetic underpinnings of testosterone levels at different ages, using the ABCD data and UK biobank data. And she's hoping to look at those in relation to male bias disorders and phenotypes. And I also want to just always thank the Turner syndrome Society of US has been very supportive of our TS studies. This is our Team and Collaborators. I tried to call out key people as we went through, but this is my full lab here at MSU. I think all of them were mentioned except for our 3 newest members, which are 2 new PA students, Kanal Patel and Monish Mayal. We've got a high school student joining us from Okemos High School as well, Alexa McCursey. Some of my wonderful MSU collaborators, Gustavo Campos, very important to our urgence work. Rob Quinn supporting our microbiome studies. Linda Mansfield has been wonderful, as well as AJ and Adam in the preclinical studies. Called out Nicole, another person from, I should say, the new grants leveraging the March cohort. That's where we'll recruit from. So Sarah Comstock's been an important part of looking at the gut microbiome in March. I've talked about most of my UNC collaborators, and you've also got a list of origins members as well. So I think